All right, next part of the endomembrane system is the vesicles. And I'm going to point those out to you real quick on here. So the secretory vesicle right here, okay, it's pointing at these two little guys over here, and right here. Okay, that's what we're talking about here, these vesicles. Okay, again, what is their function? They can store materials, they can transport materials, or they can help to break down or digest products that the cell makes that it doesn't need, or other waste products. So it's a little compartment. It's got at least one lipid bilayer around it to either move stuff around, store it, or to digest it, to break it down. They can be made in different ways. They can be made by the Golgi, by the endoplasmic reticulum, or even from little parts of the plasma membrane pinching off. Okay, all of those things, the Golgi, the ER, and the plasma membrane, are made out of lipids. So all they have to do is pinch off a little bit of the lipid, and now they've made a vesicle to move their products around, or to get rid of waste, or to bring something in. The vesicles have basically two functions as far as the direction they're moving. They could bring in materials from a different cell, or they can release materials from the cell that they're in. And then, of course, as I said, they can store materials and get rid of waste as well. Okay, the second part, maybe we're on the third part now. Third part of the endoplasmic, or of the endomembrane system, is the endoplasmic reticulum, or the ER. Its function is kind of a variety of things, because we have what's called a rough ER, that's this RER right here. And then we have a smooth ER, which is the SER. And RER is called rough ER because it has little proteins on it. And that's what we're seeing over here, or sorry, little ribosomes on it. That's what we're seeing over here. You can kind of see the little bluish bumps on the endoplasmic reticulum. Those are little ribosomes that are attached to it. So it looks rough. What happens in the RER is proteins are made. And that makes sense because that has ribosomes on it, and ribosomes make proteins. But not only are the proteins made there, but then they're also processed. As you know, proteins start as a polypeptide chain, but then go through various stages of folding in order to become that final protein. Well, it's the RER where all that folding happens. The smooth ER, which you can see a little bit of over here, okay, doesn't have any ribosomes on it. So it's not making proteins because it doesn't have any ribosomes. Instead, it makes phospholipids. See right there. Remember what phospholipids are? Part of the cell membrane and other membranes. And it can also package proteins into vesicles. So this is where we can make one of those vesicles. Here's one right here that can travel throughout the cell. So it can put the proteins into little compartments so they can move around the cell. So the overall function of the endoplasmic reticulum, taking those both into account, both the RER and the SER, is it makes proteins, it makes lipids, and it can deliver things around the cell. Okay, next, the Golgi apparatus, still in the endomembrane system here. The function of the Golgi, again, remember it's just a series of membranes, is to package, modify, and transport materials to different locations inside or outside of the cell. So once it gets a protein, so I'm right here, so make sure where you're following along here. Once it gets a protein, for example, from the smooth endoplasmic reticulum, it then basically puts like labels on it. So that's what I mean by packaging it. It puts little carbohydrates on it that says this protein needs to go to be used in the cell, or this protein needs to be shipped out to a muscle cell. So it kind of sorts out where all the proteins are going to go, and then as it says, it can distribute them within the cell or export out of the cell. And usually it does that with those vesicles. Okay, so after it packages all the proteins up in vesicles, can send them on their way. Another thing it can do is it can wrap up enzymes in a little membrane, and those membranes full of enzymes are called lysosomes, which we'll talk about in a minute, what those are all about. Okay, so here we have our lysosomes. We'll just look at the diagram first to begin with to make sure you kind of see this visually before we write it down. So on this diagram, you can see we're going to start with the rough endoplasmic reticulum down here. Okay, it's made some enzymes. Remember, enzymes are made out of protein. Okay, so it's exported some protein in a little vesicle. These proteins happen to be an enzyme. That vesicle travels to the Golgi apparatus, where the Golgi apparatus labels it and sorts it and says this is a lysosome. So the lysosome right here, which contains all those enzymes, now has a label on it, and so the cell knows what to do with it. We have what is right here, a type of food waste, or vacuole, sorry, a food vacuole that's come in from the cell. Okay, so you can see its path right here. Food comes into the cell, but now the cell needs to use that food. It needs to break it down. We're not talking about food like a chunk of chicken here, okay? What we're talking about is maybe a protein or an amino acid, something small after your digestive system has gotten done with it, and we need to break it down further. So the lysosome right here fuses with that little vacuole, that membrane-bound container of food, and it starts to digest it, which is happening right here. So it breaks it down into smaller parts. And then finally, the other thing that it can also do is a lysosome can take care of a damaged organelle. So let's say there's a mitochondria that is no longer working correctly. The lysosome can fuse with that and then also break that down. So its basic function is to break stuff apart. Okay, it contains enzymes that breaks things up, whether it's food that we need to get broken up into smaller pieces or destroying things that we don't want in the cell. Alright, so now that you kind of see the visual picture of what's happening here, let's go on to what you might want to write down. So its overall function, as we said, is to break down the food into particles that the rest of the cell can use, or to destroy old cells or cell parts, or even sometimes like bacteria and stuff, bad stuff that gets into the cell. Again, the vesicles are produced by the Golgi, so the Golgi apparatus has received these proteins from the endoplasmic reticulum, put a little signal on it, usually carbohydrate, that says this is a lysosome, and sends it on its way. And once again, in review, they contain enzymes, so they can break stuff down, basically. So they're enzymes that take a substrate and breaks it down into multiple products.